Presentation. Now it's my great pleasure to introduce my colleague and longtime collaborator, Dr. Towia Libermann, who is associate professor at Harvard. He is the director of Genomics, Proteomics, Bioinformatics, and System Biology Center at the Beth Israel Deaconess Medical Center. She's also the director of the Proteomics call for the Dana-Farber Harvard Cancer Center. Dr. Libermann, thank you for joining us.
Thank you very much. And let me just share my screen. Can everyone see?
Yes.
Yes, so I will talk today about pre-diagnostic plasma proteomics biomarkers for earlier detection of liver cancer. And I just want to go back into the explanation with regard to hepatocellular cancer, that the key characteristics are really that typically there's late diagnosis, poor prognosis, and there are limited treatment options. And it is the second leading cause of cancer death worldwide and will be the third leading cause of cancer deaths in the United States by 2030. Most liver cancers are detected at an advanced stage with a life expectancy of less than 12 months, and there are no specific symptoms for early stage hepatocellular cancer. Most importantly, liver cirrhosis patients are a high-risk population of need for earlier detection, and that includes both whether or not it's viral, alcohol-induced, or an ever increasing number of NASH patients who develop liver cirrhosis and are at an increased risk of development of hepatocellular cancer. So there's a need for blood tests to detect liver cancer in individuals who show no signs of cancer. And in order to do that, really, we need to identify a diagnostic for earlier detection of hepatocellular cancer, but unfortunately, none of that exists at the current moment. So what is needed really to detect early stage small tumors? So we need biomarkers for these early stages. We need a screening strategy for cancer in asymptomatic, so-called healthy people. And we have to develop a non-invasive test in pre-diagnostic specimens to detect early stage low abundant biomarkers. And the problem is obviously that currently there's a lack of biomarkers that can identify early stage small tumors, even so early intervention is the best chance for a cure. And there's definitely a pretty big window of opportunity because cancer development takes probably 10 to 20 years until it reaches the full-blown metastatic cancer. And if we could intervene at the earlier stage, we would definitely have a much higher chance for a cure. The challenge is obviously that small tumors release small amounts of tumor-specific proteins. And so the question is, can we use proteomics to detect such biomarkers for early detection of cancer, in particular in this case for hepatocellular cancer? The challenges in protein biomarker discovery, in particular in blood, such as serum and plasma, are that proteins or extracellular vesicles derived from the tumor tissues and cells are released into the circulation and are then diluted with all the abundant blood proteins. And most likely the biomarker proteins are present at relatively low concentrations, and the smaller the tumor is, the lower the concentrations will be due to these dilution effects. And this challenge is represented also that there are 12 logs of dynamic range in protein expression in the blood. So in order to get to the low abundant proteins such as cytokines at low picogram per ml, we have to cross around 12 logs of dynamic range. So we need a high sensitivity technology that detects few molecules of protein biomarkers. And we have to be able to measure simultaneously a broad range of low abundant proteins if we want to detect things in a relatively unbiased approach. So the platform that we are using is a SomaScan platform from SomaLogic, which is a proteomics platform that's based on modified aptamers. Aptamers are DNA oligonucleotides that form three-dimensional structures that are similar to antibodies and have high affinity and high selectivity for the proteins that you select them against. So the SomaScan platform is a high multiplex, sensitive, specific, and quantitative proteomics tool that is also high throughput, so we can run 85 samples per run, and we can measure in the older version that we have used in our preliminary data 1,305 proteins, and the latest version measures over 7,000 proteoforms for 6,596 proteins in very small amounts of serum, plasma, urine, CSF, or proteins extracted from tissue, biopsy, cell lysates, exosomes, etc. So we need very small amounts of material. We can run it in high throughput, and we can run it across a large number of proteins. It's actually currently the most comprehensive proteomics platform available. Most importantly, the dynamic range is far better than mass spectrometry and can capture proteins at low picogram per ml, such as interleukin-6, while at the same time measuring also apolipoprotein and albumin. Importantly, also the reproducibility is very high with around 5% median CV, and the platform covers the majority of the biological pathways. Our center now offers this 7K SomaScan service to any investigator who is interested, and obviously I would be very interested to talk to some of the speakers at this symposium, because they might have access to the type of samples that we are really interested to explore. So to discover now non-invasive early detection protein biomarkers in hepatocellular cancers, it's important to look at pre-diagnostic plasma from either large population cohorts or clinical cohorts of liver cirrhosis patients. However, most current studies have typically looked at early detection of cancer based on identifying biomarkers that discriminate between cancer and healthy, rather than population cohorts with archived blood that enabled the identification of biomarkers in asymptomatic healthy individuals, potentially years before cancer diagnosis. So our strategy is shown here, and I will show you the kind of data that we have generated with our first set of samples. So we are using two large population cohorts, the Nurses' Health Study and the Health Professional Follow-up Study. We take the blood plasma from healthy individuals that were archived with more than 20 years of follow-up data and diagnosis of hepatocellular cancer between 1 to 24 years after blood was collected. We then, in our discovery phase, performed SomaScan using actually, in this case, the older version of the 1305 proteins. And we overall analyzed 56 patients who later on were diagnosed with hepatocellular cancer, 35 patients who were diagnosed with liver cirrhosis, and 55 patients who stayed healthy over a prolonged period of time. And we went after detecting protein profiles that are associated with hepatocellular cancer. We then performed differential expression analysis, and we identified 59 proteins in the nurses' health study and 37 proteins in the health professional follow-up study that were statistically significantly associated with hepatocellular cancer. We performed principal component analysis and various other analyses. And most importantly, then we developed a predictor model for hepatocellular cancer and also analyzed the biological pathways using systems biology that are associated with these differentially expressed proteins. Then we selected proteins for further validation using gold standard ELISA. So we took 4 out of these 13 proteins from our predictor, and based on various criteria, how we selected these 4 proteins, and verified these proteins by ELISA on overall 146 individuals, and performed single protein quartile analysis, single protein logistic regression models, and multi-protein logistic regression models, and are working also on machine learning models. And then obviously we will further validate them on various other cohorts in order to validate independently that these proteins are indeed associated with hepatocellular cancer in pre-diagnostic blood samples. The data are shown here. First of all, for the SomaScan data, and I show just on the right side for one of these proteins, we indeed found various proteins that are differentially expressed, and you can see on in this box whisker plots on the left side in the nurses' health study, one of the proteins that is relatively low in healthy individuals then is somewhat increased in individuals who developed cirrhosis, and even further increased in individuals that developed, were diagnosed with hepatocellular cancer. And similar, we can see increased levels of these proteins in individuals who were diagnosed with hepatocellular cancer in the health professional follow-up study. And a logistic regression of this protein shows you that indeed the odds ratio, whether or not it's liver cancer versus normal or versus cirrhosis in the nurses' health or health professional follow-up study, we've seen increased odds ratio compared to the healthy or the cirrhosis patients. The proteins that were identified are enriched in various plausible pathways associated with cancer, inflammation, NAD biosynthesis, complement cascade, and coagulation pathways. We developed a support vector machine-based prediction model that includes 13 proteins and tested it on both the nurses' health and the health professional follow-up study individuals, and indeed were able, with this 30 protein model, to predict with a high accuracy of 84% in the nurses' health study and 90.5% accuracy in the health professional follow-up study. And similarly, the AUC is 0.877 and 0.966 for nurses' health and health professional study, respectively, showing that indeed this model is already quite good, maybe not as high as we would like it to be, but it's a good starting point. And when we look at more samples and go from the 1,300 proteins to the 7,000 proteins, we hope to get a much higher accuracy in AUC that is clinically useful. We, as I mentioned, use the SomaScan platform for discovery, and then ELISA for validation of biomarkers. And what is important also is when we look at ELISA and SomaScan data, we get a very high correlation for the majority of the proteins that we have analyzed so far by ELISA and SomaScan, as shown in this correlation analysis. The data for the 4 proteins that we selected out of the 13 protein predictor model are shown here in the box whisker plots. Indeed, all the 4 proteins, as they were increased in the SomaScan data, were also increased in hepatocellular cancer as compared to cirrhosis in healthy controls, with statistical significance when measured by ELISA. And when we performed a single protein quartile analysis of these 4 proteins, in this case showing you across the Nurses' Health study, indeed, we can see also an increased odds ratio for quartile 4 versus quartile 1 for all 4 biomarkers. Some of them, such as protein 4, is even showing a dramatic increase in the odds ratio, even when we take quartile 2 and quartile 3 versus quartile 1. Most importantly, when we perform multiprotein logistic regression model development of the 4 proteins, we see, first of all, for the single protein models, we see some reasonable AUC for each of the individual proteins. But when we combine the 4 protein into a prediction model, we get an AUC of 0.788, whereas the baseline model that's using age, smoking, BMI, and aspirin use only achieves an AUC of 0.644. Combining the baseline model with the 4 protein model further enhances the AUC to 0.823 for this 4 protein model. And obviously it's only 4 proteins out of the 13 proteins. So we hope once we have validated all the 13 proteins and go into various additional cohort studies, that we can achieve a significantly higher AUC from that 13 protein model. So the promise of a new test for early detection of hepatocellular cancer is that we can go from the standard of care, where in late stage hepatocellular cancer, the 5-year survival rate is between 3 to 12%, to an earlier detection with an early detection test that would then increase the 5-year survival to potentially 35 to 70% based on current data, using an early intervention that would be the best chance for a cure. So there's clearly an opportunity to develop a blood test to shift detection to an earlier stage and improve survival. And to summarize the data, the SomaScan proteomics is an effective tool for discovery of novel risk and early detection pre-diagnostic plasma protein biomarkers for hepatocellular cancer. Pathway analysis of the proteins that we have identified so far links these biomarkers to various plausible hepatocellular cancer etiological pathways. Elevated levels of multiple proteins identified in pre-diagnostic plasma years before diagnosis can predict the risk of developing hepatocellular cancer. And we have used already immunoassays to further validate 4 of these proteins across both the nurses' health and health professional follow-up study, and will now continue to further analyze them in independent cohorts, including not only population cohorts, but hopefully also longitudinal liver cirrhosis cohorts. And we will then further validate or develop additional multi-protein predictive models for earlier detection of hepatocellular cancer that will have to be further validated, obviously, for clinical utility. Thank you very much. And I would just like to acknowledge that all this work was in close collaboration, obviously, with Zhuhong Zhang and his team and various people in my lab, as well as Long Go as a biostatistician, Michelle Lay, who is a hepatologist at the Beth Israel, and Christopher Denford, who is a hepatologist at the Intermountain Healthcare in Utah. And both Michelle Lay and Christopher Denford are enrolling liver cirrhosis patients for longitudinal surveying and collection of both data and samples. Thank you very much.